Okay, guys, so we're working our way forward in laboratory now into Module 2. Up to this point, we've been looking at a lot of these kinds of plates, these kind of clear plates. These are complex media, and we've been using these plates to just look at basically macroscopic colony characteristics. So we're looking at color, odor, and so on, stuff that was in the quiz that you took earlier this week. Now, this is still important. This is still important stuff, but there's limitations, right? So what we did is we went onward and we did some staining. Okay, so we're looking at some microscopic structures. We're able to tell gram-positive from gram-negative. We're able to tell cell shape and arrangement. But now we need to go further because this doesn't really get us to the exact species when we're looking at these. So what we need to do here is move into other types of media. Okay, so here we have all TSA plates. Okay, so TSA plates are all tryptase soy agar plates. These are all a complex medium, which means that they have a variety of different nutrients, lipids, proteins, carbs that will all help. Just a majority of things will grow on these plates. Not everything, but lots of stuff. Okay. So this is going to show general growth. We're going to look at these colony characteristics. Now, if we were to take some further steps to look at what do these organisms do metabolically, what enzymes do they contain that allow them to function, we wouldn't want to forge ahead with a mixed culture. We want a pure culture. So just looking at these various plates that we see in front of us, I would immediately say, well, we need a little more work with this plate that I X'd out in green, because I see two different species here. We want a pure culture before we go on and look at their metabolic abilities, because we won't know which organism, if we're not careful about picking an individual colony here, and maybe pick up two of these species, we won't know which one actually is responsible for that biochemical characteristic, for that metabolic ability, right? Also up here I see some kind of off-white colonies kind of mixed in with these off-yellow colonies. So I'd want to do a little more work with my streak plating to get a pure culture. So the first unique plate that I want to introduce to you guys is a blood agar plate, and it's just that. It's a plate that has blood in it. Kind of gory, kind of gross to think, right? But there is 5% sheep's blood in this plate over here, this BAP plate. And so it looks very different than a TSA plate. And this is often used for organisms that are really picky about their growth conditions. They need blood to actually survive. We call these kinds of organisms fastidious organisms. And to give you an example of one of those that we often are looking for using blood agar plates, Streptococcus would be one of them. Okay, the genus group of Streptococcus. Now, what we do is we refer to a blood agar plate as not a complex medium that contains lots of different things, but more than that, it's an enriched medium. It has this very special thing, which is the sheep's blood. And we also say that this is a differential medium, which means that when organisms grow on this plate, they will show their abilities, their characteristics differently when they grow. Okay? We refer to these organisms based on how they can break down the red blood cells in that plate. We have gamma, alpha, and beta hemolysis. Hemolysis is the term for breaking down the red blood cells. You might have heard maybe in anatomy and physiology about hemolytic anemia. This is a condition where the red blood cells are broken down in a person's system. So we're actually looking for an organism's ability to produce enzymes called hemolysins. Hemo and lysins, right? Hemo for the red blood cell, and lysis or lysins for the proteins that are going to break down that red blood cell. Okay? So here we have a nice pictorial here showing us, and they actually used the sterile instrument and drew on the plate here, on this blood agar plate, what these various organisms are capable of. So they picked 3 different organisms from pure cultures, and they drew on this blood agar plate, right? So over here at the bottom, we have the Greek symbol for gamma, okay? And what gamma indicates is no clearing, which means no hemolysis. So what are we looking for? We see the growth here, right? We see the growth kind of in an off-white here, but I see just the same color, the same kind of dark red around that growth. There's no indication here that anything happened to the blood that gives it the red color. And so we would say there is no clearing, there are no hemolysins being released must be here, and so we say it's gamma hemolysis, no hemolysis. All right? All right, let's move up here to the upper left side of the plate. We have kind of a greenish bruised look. It's kind of a greenish, sometimes brownish look to the medium underneath and around the growth. And this greenish halo tells us that it's alpha or partial hemolysis. Okay? So these these alpha hemolysins only partially break down red blood cells. And this is the greenish color that we get. Okay, so we'd say this is alpha hemolysis. Okay, lastly, we might look and see that there's a translucence, an actual clear halo around the growth. And you can see right through this sometimes. I'll show you another image here in a second to kind of illustrate this. But this is called beta hemolysis. Okay? This is complete breakdown of the red blood cells anywhere near the growth. And that's why you see no red around this beta symbol. Okay? So let's look at some other images here just to kind of make sense of this. So let's look at this top one up here. We have this kind of a whitish colony here, and I see 0 clearing around these white colonies. I don't see a greenish color. Maybe it's a little off over here on the left, but really when I look at these individual colonies, there's no green or kind of a bruised look around these colonies. There's no clearing around these colonies. We would say that this is gamma hemolysis. Okay, we move down to the left here, to the bottom left corner. And here I see a very obvious clearing that happens to be around these colonies. This has to be a beta hemolysis going on. And now that we're looking at it, I just noticed, I also see here, we'll circle it in purple here, or point at it in purple. I see a little contaminant here. They must have had some problem with aseptic technique, right? So that little guy, bacterium, kind of snuck in there during the process. All right, lastly, let's look at this one. We see we have some greenish kind of halos here. Well, this would be a good example of the alpha hemolysis that we're talking about, this partial halo, this partial breakdown of the red blood cells. Okay, and finally, let's do a few trickier ones here with the blood agar plate and call it quits for this particular clip. So here what I see, up on the top left, we see some cells here have a nice clear halo, some colonies, I should say. And some of these colonies have kind of a bruised greenish or brownish look. So what I see here is definitely not a pure culture. It looks to me like it was a mixed culture, and they have a mixture of what would be alpha and beta hemolysis going on. Down the bottom left, again, we have a bruised look around some of these colonies. This bruised look to me, here's some more down here, would be again alpha. Anything like this, well, these have no halo at all. I'd say that's got to be gamma hemolysis. And the last thing I just wanted you guys to see, one of the things that's kind of like a hallmark of beta hemolysis over here, we can actually see through beta hemolysis if it's covering enough of the plate that that you can see right through it. You can read text through the plate. All right. All right, so let's call that quits, and we'll move on to some of those other media next time.